Hey there, welcome back to Biology class with Professor Tracy. Today we're going to talk about mitosis, and I just want to do a little demonstration. I know you guys have slides to look at and some videos, but I'm also getting bored and I need to do something. Alright, so here's our cell. And inside our cell, we know that we're going to have a nucleus, and inside that nucleus we have our DNA. When we begin to go through the cell cycle, as we know what happens, then we're going to go through several phases. First, we're going to be in interphase, and during interphase, we have G1, S, and G2. During G1, our cell is just growing. It's a happy-go-lucky growing cell, just minding its own business. When we progress into our synthesis, or S phase, what happens during our S phase is that our DNA is still within our nucleus. It's kind of all bunched up in there, but it's going to replicate. And when the DNA replicates during our S phase, we're just duplicating the DNA. We're not doing anything with what's going on out here in our cytoplasm. We just are replicating our DNA. After we finish our S phase, the cell goes on into the G2 phase. During G2, the cell is going to continue to grow, but it's also going to be producing extra copies of things like the mitochondria or other organelles that might be present in the cell, lysosomes, ribosomes, ER, as well as the rough and the smooth ER, or chloroplast for a plant cell. After the cell finishes doing all this replication, kind of packing up, getting things ready to divide, we have now finished interphase with G1, S, and G2, and we're going to move on to our mitosis phases. So the first phase of mitosis is called prophase. During prophase, this tangled web of DNA here in the center is going to start to form those little X-shaped chromosomes that we looked at. Each X-shaped chromosome is going to have a sister chromatid. The sister chromatids are the ones that match up to each other. So my two red fish and my two orange fish. And we're also going to see our nuclear envelope dissolve during our prophase. After prophase, we have a phase called metaphase. During metaphase, the centrioles that formed at the start of this process are going to migrate to the opposite ends of the cell. And what's going to happen is that our chromosomes are now going to line up here at the middle. So meta means middle, and they're going to be attached at the middle of their chromosomes to a spindle fiber. These spindle fibers are linked back up here at opposite ends to our centrioles. These spindle fibers are providing some directionality. So here we're at the end of metaphase. We're going to take a brief pause, and then now we're ready to begin anaphase. During anaphase, this is like the action step of mitosis. During anaphase, what we're going to have happen is that our centrioles here at opposite ends or poles of our cell, they're going to begin like doing a Pac-Man chomp. So they're going to be chomp, 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 eating away at our spindle fibers. As they are eating away at the spindle fibers, they're going to pull with them the sister chromatids and separate them. So just because my plate is cluttered, I'm going to get rid of all of that. We have now finished anaphase. We have half of our chromosomes on one side of the cell, half of the chromosomes at the opposite side of the cell. It's also very important to note that we don't have it looking like this. We don't have all of chromosome orange on one side and all of chromosome red on the other, because the information in the DNA here is different than the information on the DNA molecule here. So we need to have one copy of each chromosome on either side of our cell so that our new cells have the same DNA content as each other. So now we have finished anaphase and we're ready to begin telophase. With telophase, we're going to have our cell membrane has expanded. So our cell membrane is now really big. Our chromosomes are opposite halves of that, and we see a little pinching inward here of our cell membrane. And this pinching inward of our cell membrane during telophase tells us that the cells are about ready to actually divide. After telophase is complete, we're going to begin cytokinesis. Part of telophase also is the reformation, or starting to reform, our nuclear envelopes. And when we get into cytokinesis, this little pinching inward is going to completely pinch and separate our two new cells that are now identical to each other. It's like we've made a photocopy of this cell in producing this new cell. Thanks for listening. Hope you all are doing well. Bye.